Welcome to the THOR Table of Knowledge podcast, where we dive deep into the groundbreaking world of trauma care, hemostasis, and oxygenation research. A special thank you to our sponsor, Velico, for their support. Velico is revolutionizing trauma care with their innovative frontline ODP spray-dried plasma system, with the goal of empowering blood centers worldwide to produce dried plasma for transfusions.
Phil, let's talk about hemostatic resuscitation. Why are we even talking about this? Why is it important?
Well, it's important because patients with hemorrhagic shock develop what we've been calling blood failure, right? And that's reduced oxygen delivery that then leads to a coagulopathy, endothelial dysfunction, and also immune dysfunction as well. So the balanced, a balanced resuscitation that it simultaneously addresses the shock as well as endothelial hemostasis and immune function will very likely improve outcomes. And that's in contrast to the previous resuscitative approach where we were using crystalloids as the primary initial resuscitative solution, which in retrospect worsened outcomes by causing a dilutional coagulopathy, causing increased inflammation, increasing endothelial dysfunction, and worsening hemostasis. So we're talking about hemostatic resuscitation because moving forward, it appears to be an optimal approach compared to the classic approach, which was crystalloid based.
Right. And in fact, I mean, we can look back through history to early indications that patients were developing problems with coagulation function. And we saw reports of this in Korea and Vietnam. Famously, Gene Moore and colleagues described the lethal triad, right? Caused by crystalloid resuscitation, so giving a lot of saline, causing acidosis, hypothermia, and dilutional coagulopathy, very bad, not to mention diluting the red cells. So we kind of knew that we need to do something better, right, for a very long time. I think one of the interesting developments over the last few decades was the description of what we now call, by many names, trauma-induced coagulopathy or acute coagulopathy of trauma. It's had sort of different iterations, but this idea that even before you pour saline into people and dilute the blood, there are alterations in hemostasis function detectable through standard coagulation assays. So Karen Brohe and Mitch Cohen described this in the early 2000s from their work at UCSF, showing that in patients, even before resuscitation started, had increasing INR, International Normalized Ratio, and prothrombin time. They had documented that factor V and factor VIII dropped a little bit. They hypothesized that maybe this is sort of an anticoagulation response driven by protein C. But while this is all happening, people were sort of thinking about resuscitation differently, as you started to say, even without really focusing on the anticoagulation, but knowing that, boy, everything seems to be failing at the same time, right? And we need to give blood back. So tell us a little bit about how that, I think today we talk about hemostatic resuscitation with a view towards resuscitating coagulopathy, but that's not actually how, quote, balanced resuscitation came about. It was really more in response to the saline bit, and a lot of that had to do with your experience in training and then in Iraq. Why don't you talk a little bit about the birth of that?
Right. So interestingly, many people do think 1:1:1 came before whole blood, and then we transitioned to whole blood after learning that 1:1:1 was associated or with improved outcomes, and it's the opposite. We started in Baghdad using whole blood to avoid using crystalloids and at the same time provide a balanced resuscitation. Whole blood came first. While for the time it took to collect the whole blood, the 30 minutes or so, we said, well, until we have whole blood available, we should at least give 1:1:1 before the whole blood can be utilized.
To kind of mimic whole blood, basically.
Yes, to mimic it or to reconstitute whole blood with 1:1:1. So that's how we started using 1:1:1. And it's interesting, as things got very busy in Baghdad in 2004, we actually went to Saddam Hussein's library on the second floor of Ibn Sina, which was his hospital in the center of Baghdad. And I remember vividly, we went into his library and the 8 of us sat down and wrote out a protocol that Tom Repine eventually published later the next year, where we said, well, while we're waiting for whole blood to be collected, we're going to proactively, right, give 1:1:1 until we have the whole blood available. And again, the goal there was to provide a balanced resuscitation that simultaneously addressed shock as well as the coagulopathy. Now, we really didn't understand the endotheliopathy yet. That came later, right, as we started to think about, well, why is plasma potentially improving outcomes? And it was, Shabani Pati and Rosemary Kozar that have really led the way in first describing the endotheliopathy of trauma and understanding that aspect of it. But 1:1:1 was a bridge to whole blood to provide a balanced hemostatic resuscitation.
And actually, the Armed Services Blood Program struggled with providing platelets early on. It was only really in a few locations that platelets were available, and that caused a lot of heartache about resuscitation. Why don't you talk a little bit about that?
So you're right, and because I was there in Baghdad in 2005, because the blood establishment back then was not happy that we were using whole blood, because at that point, and still with using warm, fresh whole blood, it's not formally tested for transfusion-transmitted diseases. We were using a rapid infectious disease screening kits that we bought from Spain that would rapidly test for HIV, Hep B, and Hep C, but it wasn't to the FDA standard. So there were many blood bankers, very, very worried about that risk. So what they did in January of 2005 was they sent platelet machines to Baghdad to try to prevent us from using whole blood. The concept from their perspective was, well, if we give you the pheresis machines and we send a tech out that can collect platelets for you, that will stop you from using whole blood.
Right.
It didn't. We continued to use whole blood, but we did start to collect platelets and then give, again, components in a 1:1:1 ratio until whole blood was available.
So, all right, while all this is happening, Mitch Cohn and Karen Burr here publishing their work on coagulopathy of trauma, but that really didn't influence what you guys were doing. You guys were just trying to provide blood because you knew that that's what the patients were losing, and it was better than giving saline, right? Now, later on, we get the data, right? So first was the 2007 plasma to red cell ratio paper showing that patients who got something close to 1:1 did better than patients who got sort of a very red cell heavy and saline resuscitation, right? Then comes the paper that says adding platelets, Jeremy Perkins and colleagues, also improves outcomes. Then we had civilian data, right, from the PROMPT study, observational study, showing the same thing: more platelets, 1:1, better than just red cells kind of resuscitation. And then the PROPER study, which was a randomized trial comparing 1:1:1 versus 1:1:2. Interestingly, in that study, there was supposed to be a third arm, 1:1:4, so much more red cells in relation to plasma and platelets. But by the time that study was ready to start enrolling, nobody would randomize patients to 1:1:4 because of all the other data, right? Equipoise was lost. So what's interesting too about the PROPER study, although it was technically negative in the sense that the 95% confidence intervals overlap between the two arms, there was an increase in early survival with the
reduced death from bleeding at 24 hours,
right,
and increased time to hemostasis with 1:1:1 compared to 1:1:2.
And if you look at the patients who got the per protocol analysis, Jessica Cardenas' Blood Advances paper, like the people who actually got the platelets when they're supposed to get the platelets, the mortality difference was huge. It was like a 20% difference. So the idea of replacing the parts of blood that provide hemostasis, when I talk about hemostatic resuscitation, the idea that putting those back into the bleeding patient is better for the patient has turned out to be empirically true in both the observational studies and then later the randomized trial. But here's the crazy thing. Are we really fixing coagulopathy? And this is where, I think it's interesting to go back to the coagulopathy of trauma literature, because it all started with this observation that the INR, or the prothrombin time, is increasing. And the assumption was, hey, just like patients on warfarin, these bleeding patients can't generate enough thrombin to clot the blood, right? But actually, when we go back and look at thrombin generation in these patients, and a lot of this work done in Houston by Charlie Wade and colleagues and John Holcomb, you see that actually thrombin generation's up, right? Now, platelet function seems to be altered, and a number of different groups have published that, including the UCSF team and Karen Brody's group in London. Fibrinogen seems to go down, but really the diagnostics give us a confusing picture. We don't actually know what the etiology of all these changes might be or how to fix it. So we talk about things like precision medicine and the need to do, quote, hemostatic resuscitation. But it seems to me like what we're really kind of doing here is a first do no harm approach, right? Don't give them saline, which causes demonstrable harm. Give them back what they're missing.
Correct. And there's randomized controlled trial data, supporting the fact that crystalloids are harmful. A secondary analysis of the Pamper trial, published by Jason Sperry, clearly showed that the patients receiving only crystalloids had by far the worst outcomes compared to patients that received either only red cells or only plasma, and those who did the best in that trial got a combination of red cells and plasma. So there is RCT level evidence showing that crystalloids are harmful. Completely agree with hemostatic resuscitation. One of the main principles is, again, doing no harm. And you're right, our current assays don't help us really better understand the pathophysiology. We as a community need to engage more with our bioengineering colleagues and figure out more accurate ways of developing in vitro assays that replicate in vivo hemostasis, as well as shock and endotheliopathy and the immune dysfunction that occurs too. Because until we better understand the problem, it's going to be very difficult for us to develop more precise solutions to that problem. But at least currently, the big advance we've made over the past 20 years is giving less crystalloids, giving a more blood-based approach. The future is going to be providing individualized or personalized care based upon the type of coagulopathy and shock and endothelial immune dysfunction that you develop versus myself. That's the next frontier as we work towards improving outcomes for patients with hemorrhagic shock.
So quick question for you. You mentioned our bioengineering colleagues. I want to give a shout out to Susan Shea and her work on microfluidic systems, which help us get closer to what's actually happening in vivo than some of these static assays that we're currently using. I think it'll be interesting to see how the results from those assays correlate with clinical trial data. And we're getting some of this from the CHIP study, right, on cold platelets. Do you want to quick mention how we're doing this sort of in vitro, in vivo correlation that might help us understand?
Yeah, definitely. There's a big move in developing more accurate microfluidic assays that will more closely represent in vivo hemostasis. And what's important or different about the microfluidic assays, right? It incorporates flow into the assay as well as biologic surfaces. So in the models that Susan Shea has developed and others are also started to develop, you can alter flow, right, to represent venous flow or shear right, versus arterial. And because we know flow is very important to activating the hemostasis, as well as biologic surfaces within these microfluidic assays, whether they're tissue factor or collagen impregnated. It more accurately represents, again, in vivo hemostasis, whereas our current assays don't incorporate flow. They don't incorporate biologic surfaces, which can lead to either over-representing hemostasis, which I think the viscoelastic assays do, or under-representing hemostasis, which I think the aggregometry-based assays do. So in the CHIPS trial, as well as now other trials that we've started, we are also examining hemostasis in those subjects, as well as based upon the products themselves with these microfluidic assays to better understand if how you make the blood product, right, or how the patient responds to different resuscitative approaches affects hemostasis in a different way. Susan and others are working on endothelializing these microfluidic chambers, which will add another dimension too. So hopefully in the future we'll have more accurate assays to better understand the pathophysiology as well as the response to different transfusion therapies.
I guess that'll be for future podcasts. Our audience can tune in as we explore new data on better diagnostics for coagulopathy of trauma and help us understand better how to do hemostatic resuscitation with precision.
That's a wrap for this episode of the THOR Table of Knowledge podcast. A big thank you to our sponsor, Velico, for supporting the advancement of trauma care. Their frontline ODP spray-dried plasma system is paving the way for blood centers globally to produce potentially life-saving plasma for transfusions. Check out our website, www.thornetwork.com, for more podcasts, and join us next time as we continue to explore the cutting-edge science of trauma, hemostasis, and oxygenation. Until then, stay safe and stay informed.